Different types of feeding habits in fishes and their adaptation in alimentary tract, physiology of digestion, respiration and osmoregulation in freshwater and marine fishes. Introduction. Fishes exhibit diverse feeding habits and possess specialized adaptations in their digestive and respiratory systems to survive in different aquatic environments. Understanding these physiological mechanisms is crucial for comprehending their ecological roles, energy acquisition, and survival strategies. This lecture will explore the different types of feeding habits in fishes, their digestive system adaptations, and physiological processes such as digestion, respiration, and osmoregulation in freshwater and marine species. Developmental events in digestive systems of fishes. The digestive system of fishes develops in response to their dietary needs. During embryonic development, the gut forms from the endoderm, differentiating into various regions such as the buccal cavity, esophagus, stomach, intestine, and associated glands. The complexity of the digestive tract varies among species based on their trophic niche and feeding behavior. Types of food consumed by fishes. Fishes consume a wide range of food items, including plankton, algae, detritus, small invertebrates, and other fishes. Their dietary habits influence their morphology and physiological adaptations, allowing them to efficiently utilize available food resources. Types of fishes based on feeding habits. Plankton feeders. These fishes feed on microscopic planktonic organisms. Example: sardines, Sardinella spp. Anchovies, Engrolidae family. Herbivorous fishes. They consume plant material such as algae and macrophytes. Example: parrotfish, Scaridae family. Grass carp, Tenopharyngodon idella. Carnivorous fishes. These fishes primarily feed on other fishes and invertebrates. Example: catfish, Siluriformes order. Barracuda, Sphaerina spp. Omnivorous fishes. They consume both plant and animal matter. Example: tilapia, Oreochromis spp. Common carp, Cyprinus carpio. Types of fishes based on food selection. Monophagic. Fishes feeding on a single type of food. Example: pipefish, Cygnathinae family, feeding exclusively on plankton. Stenophagic. Fishes consuming a limited variety of food. Example: butterflyfish, Chaetodontidae family, feeding on coral polyps. Europhagic. Fishes with a broad diet. Example: catfish, which consume a mix of plant and animal matter. Types of fishes based on trophic niche. Surface feeders. These fishes feed on floating food. Example: hatchetfish, Gasteropelicidae family. Column or mid-water feeders. They feed in the water column. Example: tuna, Thunus spp. Bottom feeders. These fishes scavenge food from the substrate. Example: stingrays, catfish. Types of fishes based on manner of capture and ingestion. Grazers. They scrape algae or biofilm from surfaces. Example: surgeonfish. Strainers. They filter plankton from the water. Example: mantare. Parasites. These fishes derive nutrition from host organisms. Example: lampreys. Suckers. They suck food particles or fluids. Example: sucker catfish. Gastrosomatic index and food quality. The gastrosomatic index, GSI, is used to assess the feeding intensity in fishes by determining the ratio of stomach weight to body weight. Food quality affects fish growth and survival, necessitating a diet rich in essential nutrients. Alimentary canal adaptations. Teeth and tongue, used for capturing and processing food. Buccal cavity and pharynx, important for food intake and preliminary digestion. Gill rakers, help in filtering food. Taste buds and mucus-secreting cells aid in food selection and lubrication. Esophagus transports food to the stomach. Stomach, involved in food storage and digestion. Intestine and pyloric ceca assist in nutrient absorption. Rectum, responsible for waste excretion. Digestive glands and their functions. Liver produces bile for fat digestion. Pancreas secretes digestive enzymes. Glands in the digestive tract aid in secretion of enzymes and mucus. Physiology of digestion. Fishes utilize enzymatic and mechanical digestion to break down food. Proteases, lipases, and amylases play crucial roles in digesting proteins, fats, and carbohydrates, respectively. Nervous and hormonal control of digestion. Nervous control. The autonomic nervous system regulates digestive processes, ensuring proper enzyme secretion and gut motility. Hormonal control. Gastrointestinal hormones like gastrin, cholecystokinin, and secretin regulate digestive activities. Adaptive modifications in digestive tract of fishes. Mouth and jaws. Modified for specific feeding habits, e.g., elongated jaws in predatory fishes. Lips and teeth. Adapted for food grasping and grinding. Gill rakers, specialized in plankton feeders for filtering food. Barbels and taste buds help in detecting food in bottom dwellers. Esophagus and stomach varies in size based on diet, absent in some herbivorous species. Intestine, longer in herbivores for plant digestion, shorter in carnivores for rapid protein breakdown. Respiration and osmoregulation in freshwater and marine fishes. Respiration. Gills extract oxygen from water. Lamellae increase surface area for efficient gas exchange. Some species exhibit accessory respiratory structures like labyrinth organs in air-breathing fishes, e.g., anabas. Osmoregulation. Freshwater fishes prevent excessive water intake by excreting dilute urine and absorbing salts. Marine fishes drink seawater and excrete excess salts through specialized chloride cells in gills. Conclusion. The feeding habits, digestive adaptations, and physiological processes of fishes illustrate their evolutionary strategies for survival in diverse environments. Understanding these aspects is essential for fisheries management, aquaculture, and conservation biology.